I know you're all waiting for me, everybody. It's just me trying to set up in a different location. I needed a hot second to set up. I haven't forgotten about us at all. Don't you worry. So I'm going to wait just a couple more seconds. Just while everybody joins. Don't forget, everybody, that if you do miss some of this or you get load shedding or anything like that, then this video gets posted onto my channel. So you'll be able to see it there. I'm also just trying to get this into focus. There we go. If the video at any point is blurry, or the sound is not great, then that potentially is one of your settings on your side. My settings are, I'm shooting on 1080p, so maybe you need to turn up your settings, your video settings on your video to see it more clearly. Okay, so I think let's dive straight into it. I think everyone that wants to join will continue to join as we go along. All right, so we're doing paper 2 stuff. And I think a good place to start is one of the most overlooked topics, because I think it's only about 12 or 16 marks, which is meiosis. And I know many of you probably think, man, this is such a waste of time. It's such a short section. But I find that many of us don't know what's going on in mitosis. And generally in exams, even though it's only for 12 marks, if you get all 12 marks wrong, you lose out on 4%, right? So I think let's quickly go over meiosis very briefly. So first of all, what is the purpose of meiosis? So there's a couple of purposes that we need to go through. We need to know that meiosis is linked to genetic variation. We also need to know that meiosis is about making haploid gametes. And we need to know that meiosis is linked to having the correct chromosome number. Now, I know that these seem very obvious things, but a lot of people don't know this list. And often the question is, what is the purpose of meiosis? And they generally just give this one over here, but they don't know the others. When it comes to genetic variation, there are 2 sources of genetic variation that we must be familiar with, which is crossing over, which I'll talk about just a little bit more now, and what we call random arrangement. Now, these 2 refer to the chromosomes. There are actually other sources of variation I'm going to get into as well. These are the 2 main sources, but there are more, right? Okay, so I'm going to touch on this genetic variation more now because I'm actually going to go into some of the phases, and I'm going to do the phases that we get wrong all the time. And I'm going to start off with the difference between prophase 1 and prophase 2. For some reason, we really suck at identifying these 2. We confuse them with each other. So prophase 1 and 2 are often misidentified because you don't know what to look for. So generally in prophase 1, you're going to have homologous chromosomes, and they are going to be partnered up like so, and they're going to go through crossing over. So when I'm looking for prophase 1, what am I looking for? I am looking for crossing over, homologous pairs. I am looking for bivalence, which, by the way, these are bivalence. This partnership here, when they're touching, is bivalence. And also, I know that I am in prophase because they haven't arranged themselves anywhere particular in the cell. They're just sort of floating out in the middle of nowhere. So there is currently no arrangement In other words, they haven't aligned on the equator. They aren't being pulled apart. The other thing I suppose that is happening in the background over here is these are the centrioles forming the spindle fibers. The problem is that in prophase 2, we misidentify it being prophase. And I want to show you why prophase 2 gets misidentified all the time. So, number one, before you say to me, ma'am, why didn't you make the green with the green and the pink with the pink? Well, first things first, that is because my pink chromosomes, and maybe I'll make a little key at the top here, my pink chromosomes are going to be my maternal chromosomes, and my green are going to be paternal chromosomes. So you can only have one of each in a pair. So I didn't put pink with pink because I didn't want to work that way. Okay. Just looking at the comments quickly, if you can't see clearly, again, that is a thing on your side. It's not a thing on my side. I do know that it's not a thing on my side also, everybody, because I can see my live video on my laptop, so I can also see what you're seeing, and it's pretty clear on my side. So it's a thing you just need to change on your video settings. Generally, if you're streaming a video, you have to change the settings because it's going to select the lower setting to save you data. That's my little piece of advice. Okay, getting back to the lesson. All right, so prophase 2. Let's get back to that now. So here what we have is we have our homologous partners that have been separated. And maybe what I'm going to do is I'm going to give these numbers. So this is pair number 1 and this is pair number 2. Now they have undergone meiosis 1 and they have separated, right? Now you'll notice that that means that, let's say this is the pink one from pair number 1 and this is the green one from pair number 2. That is because when random arrangement occurs, you get one of each of the pairs. That is a law of genetics. You cannot have both number 1s in one cell. You have to have one of pair 1 and one of pair 2. And ma'am, what law is that? That is the law of segregation. It is a Mendel law, a Mendelian genetics law, where you are only allowed to inherit one of the homologous pairs into, or you cannot, not inherit, excuse me, you can only inherit one homologous partner or one allele from each of the pairs. You can't have both number 1s or both number 2s. So number 1, I know that this is prophase 1 because of the look of my chromosomes, which are recombinant. Number 1. So what is a recombinant chromosome, man? A recombinant chromosome is just what you can see here. You can see I've added an extra little crossing over pieces where a little bit of the pink one has rubbed off on the green and a bit of green on the pink. That's because they've gone through crossing over. Number 2, I know that this is prophase 2 because there is no crossing over. Now, I get a question very often, ma'am, why is there no crossing over? Remember, they can only cross over if they are with their homologous partner. So if I go back to the original picture, if this is number 2 green, the number 2 pink is in the other cell, which means its partner is not here. Therefore, they cannot cross over with one another. Likewise with my pink number 1. Number 1 green is in the other cell. It is not here, so they cannot cross over. And because they are not homologous partners, they cannot cross over. There's no more variation that can occur through that mechanism. Okay, so this is the first reason why we get these two confused. We think that crossing over can happen in both, but they can't. And also, when we see our chromosomes like this, where they have a little extra little bit on them, we often confuse that as being, oh, it must be metaphase. But let's remember, the easiest way to identify metaphase is that our chromosomes have aligned on the equator. But yet again, We also misidentify metaphase very regularly. Let me show you the problem people have. When we talk about metaphase 1 and 2, the problem is we don't know what to look for and we look too quickly. Let me show you what happens. So here are my two cells. This is the difference between metaphase 1 and 2. The chromosomes are going to align on the equator. Generally, they are still attached to their partner. The spindle fibers or the centrioles have moved to the poles and they've grown out their spindle fibers. Versus on the other side here, this is our metaphase 2, you'll see the immediate difference when I draw it. You will notice that there is only one of each partnership here with their recombinant genes on them. And immediately, when you see chromosomes on their own like this and not paired up, you should know that this is metaphase 1, this is metaphase 2. These two phases are often misidentified. Why? Because you generally have nothing to compare it to. In an exam, they don't give you things to compare it to. Normally, there isn't lots of other phases for you to look at and go, oh, okay, I know for certain that's metaphase. Okay, so metaphase is where they meet in the middle or at the equator, but which one is 1 and which one is 2? Number 1 is where there is still a homologous pair present like this, and they're still touching or in close association. Whereas this one over here, you will notice there is only one chromosome here, and it's very soon going to be pulled apart, and these two chromatids are going to go in opposite directions. Now, the last bit of advice I want to give you on meiosis is what to call what. I know that sounds very cryptic, everybody. Like, what does she mean by what to call what? What I mean is a lot of people don't know when is it a chromosome, when is it a chromatid. So let me make it very simple. This is a chromosome. Specifically, this is an unreplicated chromosome. But this is also a chromosome. Except this one is a replicated chromosome. These two are identical to one another. That's why they still count as one. Chromosomes refer to genetic information, and if the information is the same, then there is no duplicate. So this is one chromosome, this is also one chromosome. You may only call these things chromatids if there is a centromere present. Okay? So this is a chromatid, this is a chromatid. Together with a centromere, it forms a chromosome. The moment you destroy the centromere, which means you remove it, and these two chromatids move apart, they become singular, like so, you can now refer to them as chromosomes. So this is what I tell my students. If there's a centromere, we can talk about them as being chromatids. The moment the centromere is gone, we talk about them as being chromosomes. So the key here is there a centromere? Yes, then we call them chromatids. Collectively, this whole thing is a chromosome. If we take the centromere away, you end up with two individual chromosomes. The other little bit of advice I want you to use is the word sister is not allowed in meiosis. You do not get sister chromosomes, sister cells, daughter chromosomes. Please avoid the word sister or daughter, because in biology that means identical. If something is a sister, it is a copy of its parent. And in meiosis, we know that the cells are not copies of each other. They're actually new cells. Okay, so I'm going to pause for a quick second and read your comments just so I can see what everyone is asking and saying. All right, genetic crosses, don't worry, I'm going to get into that. The random arrangement of DNA sequences by the breakage and rejoining a chromosome. Oh, I think someone is defining something. Jeremiah, please have an exact definition of a recombinant chromosome. Sure. So what is an exact definition of a recombinant chromosome? So let me quickly show you what one would look like. It would look like this, and it would have a section on it. Or it can look like this, where it's just singular. So remember, these are both chromosomes. So what is a recombinant? A recombinant chromosome and actually, by the way, I have these definitions in my study guide. I have a whole section dedicated to meiosis terminology and what it means for it to be a recombinant chromosome. A recombinant chromosome is when homologous, oh, I'm not spelling homologous now correctly. It's homologous, there we go. Chromosomes exchange genes and become different. So that's my little definition of what a recombinant chromosome is. So this whole thing is a recombinant chromosome, and this is also a recombinant. This is just a double-stranded, and this is a single-stranded. Okay. All right. I'm going to look back at the live session and just see what anyone has asked for. DNA, evolution, definitely, ma'am. Please explain bivalence and pairs. Okay, that's going to be the last thing I'm going to do for meiosis, and then I want to move on to DNA. Okay, so this is my last bit. Number one, you have 46 chromosomes, yes? And they come in 23 pairs. How are they paired? They are paired via maternal and paternal characteristics. In other words, you have a mom chromosome and you have a dad chromosome. And you have 23 of these. Okay, so let's say this was the maternal one and this was the paternal one for dad. Okay. Now, these pairs of maternal and paternal, these are homologous pairs. So what is a homologous pair? A homologous pair is a set of chromosomes that have the same size, they have the same shape, and they have, and I use the word similar instead of same, sorry, not similar shape, whoops, not similar shape, similar genes. The reason why I say similar genes is that they don't have identical genes, but they have the genes in the same space or the same loci. In other words, let's say this is the mom chromosome and mom is carrying the brown eye gene and dad is carrying a green eye gene, right? They have similar genes in that they're both carrying eye genes, but do you see that the actual characteristic is slightly different? So we say they have similar genes. So that is a homologous pair, and these are the things that you use to quantify it being a homologous pair. Then in terms of what is a bivalent, I'll show you now what a bivalent, let me make this bigger. A bivalent is when our pairs come together. So here is my maternal touching my paternal, right? So they're crossing over. This is crossing over. They touch over here. The point they touch is called chiasmata or chiasma. How did they move together? So they move towards each other via something called synapses. So that's how they moved together and touched, and they touch at chiasmata. And when they are touching like this, because they are now touching, we give them a new name called a bivalent. So when they're touching like this, that is the only time you will call them a bivalent. If they are not touching, then we just refer to them as a homologous pair. All right, I'm going to leave it there, and I'm going to move into some questions on the chat. People are asking about Down syndrome. So yes, Down syndrome is when you have an extra number 21. And everybody, I suggest you go and look at your guideline today and go and learn it. There is a definition for abnormal meiosis when it comes to having an extra chromosome for Down syndrome. Please go and learn it off by heart. That is the explanation you would use if you had to explain how we inherit Down syndrome. Also, don't overlook what creates Down syndrome, which would be non-disjunction. When these bivalents or these homologous pairs, they fail to separate and you end up having extra chromosomes. Okay, let's have a look here. Corley, I see your comment. Don't worry, I will reply to you. I do see it. I will get back to you. I'll have a look at all my emails. All right, so next thing, let's go on to DNA, right? That's going to be the next most common requested one. I actually think, to be honest with you, DNA will be probably the easiest section you will have to do. In other words, it's not going to have the most it's not going to have the most difficult application questions, so you can actually do really well. But let me show you what to expect. So let's look at DNA. Now I'm going to do a quick recap on the structure of DNA because we all need to be on the same page. So first things first, I need us to know that the structure of a piece of DNA is pretty repetitive. We have a phosphate, a sugar, and we have a nitrogenous base. There are 4 bases, which is A, T, C, and G: adenine, thymine, cytosine, guanine. And we know that these are complementary in the sense that A always joins to T and C always joins to G. That is their relationship. Why do they join like that? Because if we have a string of DNA, if the letters are A, T, T, C, then the complementary side will be T, A, G, right? Because DNA is double-stranded and it's complementary, right? So that's the basics of the structure of DNA. RNA, on the other hand, the sister to this, same structure over here, except, and this is important, the sugar in DNA is called deoxyribose, whereas the sugar in RNA is called ribose. That's the differences in their name. That's why this is DNA and that is RNA, ribonucleic acid versus deoxyribonucleic acid, okay? And the other difference here is in our pairing. A joins to U and C joins to G. So uracil is the new guy on the block. And the other main difference is RNA is single-stranded, which means that it would be one-sided like this. It wouldn't have two-sided, right? So now that we've gone over the basics about DNA, I want to go over the whole process of forming mRNA, reading mRNA correctly, and essentially this is probably the main place we would end up getting marks lost, is in protein synthesis. Okay, so protein synthesis happens inside the nucleus, and you have a piece of DNA that is wound up, right? And I'm hoping everybody is taking this description from their guideline, not from their textbook. Please use the one in the guideline only. That is what the memo will be based off of, okay? So what happens is, if I were to just eliminate this line so I can draw a little bit more in here, we unwind and we unzip a section of the DNA. So this is me unwinding this section over here. We want to unwind this section. And we have a whole bunch of nucleotides over here. And they would have their corresponding lettering on the other side, like that, right? Now, when it comes to protein synthesis, we only actually copy one side. We are not going to copy both sides. So if I wanted to create a copy of this DNA, I am going to use one side as a template only. So I'm going to use this purple side. And I'm going to use freely floating RNA nucleotides. And it's really important that you call them RNA nucleotides in your descriptions tomorrow, everybody. Don't just call them nucleotides. Differentiate between the two. Is it a DNA or is it an RNA? And these freely floating nucleotides, they complement this side of the DNA, the side we're going to copy, and they start forming a piece of mRNA. So let's do the complementary side, and I'm actually going to add on a few extra letters. So if this is A, it means it'll be U. That's T, so it's going to be A. C is G. C is G, and I actually want to add two more on here. So we've got at least 6 because I want to work in 3s. Okay, so now I have formed a piece of mRNA. Okay, that's what this piece is. And this is what I'm going to take out of the nucleus. So if this was the nucleus, the edge of it, I'm going to take this, I'm going to go out through the nuclear pore into the surrounding cytoplasm on our way to the ribosome. And this process is called transcription. Okay, right. So now this little piece of mRNA I'm going to take outside, and I'm just going to write the letters here for me so I can remember them, so I can use them in my next part of my explanation while I remove this. So now we go into translation. So here is my ribosome, and often ribosomes sort of look like a coffee bean type shape, and along comes my piece of mRNA. 1, 2, 3, 4, 5, 6, right. So I'm going to go and put my letters in. Here we go. Just wanted to keep that in the bottom here. Okay, so in comes my mRNA. And now my ribosome is going to read the letters 3 at a time. And these 3 letters are called codons. Now, these codons are often given to you in an exam in a table. And they'll say something like mRNA on this side, and then they'll have amino acid on the other side. And then they will have like GAG, AUC, etc. More options. And then they'll say like, this is proline. That's the amino acid's name. Or this is, let me think of another one. This is arginine, etc. Right? Now, when we get these kinds of questions, we need to look very carefully at the headings to make sure that it is mRNA that they're talking about. Up here, actually it's such a dark color. Let's use a different color. Over there, this is mRNA. Sometimes they like to trick you. So be very, very careful that this says mRNA. If it says DNA, what are you going to do? Don't panic. All you need to do is then convert the DNA to match the codon that you have here. So right now, GAG is a mRNA codon. It's not DNA. If this was DNA, you would have to work it backwards. In other words, if it's GAG, then the DNA code was CTC, if that was the DNA, right? So now you know the CTC, and so you can go back and you can go, oh, CTC is this protein, this amino acid. So you've got to be very careful about the headings. I would make sure that this is what the heading says. Be very clear, because they do like to trick you on that one a little bit. It's unfortunate, but it's to make sure you actually are reading the question. Okay. So let's say hypothetically, though, that it does say mRNA, and you're going to read these 3 codons, and maybe they'll say, like, what is the anticodon of this? So what is an anticodon? Well, in our cytoplasm, we have something called tRNA, which at the ends over here, like that is where we carry our anticodons, and they are complementary to the codons. And so if we match these up, then this would be the letter C to match G. This would be the letter U to match A. And this would be the letter C to match this G. And we call those anticodons. So the anticodons and codons, they match together, right? Now, sitting at the back of the tRNA is an amino acid. And a lot of people get this confused. The amino acid is linked to the mRNA, not to the tRNA at the bottom. In other words, when you read these tables, it is generally an mRNA and a amino acid. It's very rarely a tRNA at the top here. But again, remember, we must read the table carefully. Is it mRNA, dRNA, tRNA, DNA? Sorry, you need to be very, very careful that you know what you're looking at. But for the most part, it's going to be an mRNA codon table. And so sometimes they will then, like as I said to you, they will say, what is the amino acid? And we go GAG, go to the table. Oh, it's proline. And we write that down as our answer. Now let's talk about all the ways they are allowed to ask this and also confuse you. I can go into more detail about how protein synthesis works, but I do have a very good video on my playlist, so you can more than welcome to go and watch that. I think that would be very beneficial. I want to focus on the questions they will ask tomorrow and some of the issues we get. I think that's where that kind of focus is more important today. So you get a table, right? A codon table in the exam. And this side says mRNA. This side says the amino acids. And let's say they say something like a mutation happens. And the moment you see mutation, your spidey senses must be tingling, right? You must be like, ooh, you must be ultra alert. Because sometimes this is where you lose marks and you don't know what to do and you give the wrong answer. And it's because you are, let's say, fooled by the question they've asked. So let me explain what I mean by that. They give you a table. I'm just going to draw out 4 options for us here. And let's say the option is like UCA, and that gives us proline. And then let's do like UGG, ACA, and then let's do another one like GUA. And let's do arginine as an example. Let's do proline again, and then let's do methionine. And if you ask me, ma'am, do we have to know all the amino acid names? No, you don't. I just know them from learning them in varsity. So now let's say the original code. So I'm going to say here the original code was UCA. It is mutated to ACA. And the question says, what is the outcome? Now, when you're in an exam and you're nervous and you're stressed and you're working really hard, you go, oh, well, the letters have changed, right? This letter is now this letter. So clearly there must be a different amino acid. Therefore, it's a different protein and blah, 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 mutation. But you must take a second to look at the table that they've given you. Because if you look clearly, these two mRNA codons, they code for the same amino acid. And that is possible because on average, there are at least 4 codons that all code for the same amino acid. So don't always assume that if there is a mutation, there must be a change in the amino acid. You must go back to the table and double check. Okay? So double check. If there is no change like this, then all you have to do in your written answer is you mention for 1 mark what it was. You mention for the second mark what it became. But then you say, however, they both code for proline. Therefore, there is no change in the protein, right? And generally, that would get you another 3rd mark here and a 4th mark there. However, if there is a change, so let's say the mutation occurred and the amino acids were different, how do you explain that? If they say, what is the implication of changing the codon? Well, now what you need to do is, if this was different, let's say it did become something else. It became this other one. It became UGG. Again, you're going to acknowledge that it was this. It's now this. You are going to acknowledge that it is a different amino acid. But now you'll notice that sometimes these questions are out of 5 or 6. And now we're only on 3. Where does the rest of the marks come from? The rest of the marks come from your translation explanation. Now you're like, ma'am, what do you mean by that? Well, you know, when we talk about translation and protein synthesis, the mRNA is read 3 codons at a time. The corresponding tRNA joins with it, blah, 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 blah. That explanation. Now what you have to do is you have to make it in the negative. So you will say something like, the original codon was this. It has been changed to a different codon, this. Therefore, a different amino acid will come. A different tRNA will come, which means a different protein will be formed. And now you can rack up more marks by using your translation explanation, but making it negative, meaning you're not writing in the positive. You're not saying there will be an mRNA that will match with a tRNA that will bring the correct amino acid. The negative answer is when you are saying this has changed to this, therefore a different amino acid is dropped off because it's a different tRNA, and therefore we will make a different protein. Mutations are one of the most common ways they like to examine DNA and DNA replication. So I would expect one of these tomorrow. All right, I'm going to pause for 2 seconds and have a look at the comment section to see if there are any questions. I would also appreciate it, everyone, if you didn't flood the comment section with things that are not important. I would prefer if you left the comment section for questions, and maybe if you're answering each other about a question, that's great. But using the comment section to chat makes it really difficult for me to help people and see what everyone is referring to. Okay, Let's see. Somebody said there's no sound. Do we still have sound, everybody? I'm just double checking. Let me know. Claire wants to know, will we get a essay question? No, but you can get a question out of maybe like 8 marks. That would be like the top level. That would be something you would see in evolution, which don't worry, I'm going to get onto soon. Sound is good. Okay, great. I think it disappeared for a hot second there just because I think someone tried to call me, so it interferes. Lillian wants to know, can I do a need to know for evolution? Absolutely. I think that's what we're going to do. Ma'am, can you give a clearer explanation on the brief history of the discovery of DNA? I can, but can I make it even simpler for you? All you need to know about the discovery of DNA is 1, it's a double helix shape. 2, it was discovered by Watson and Crick. And 3, they used X-rays to discover the shape. That's it. That's all you need to know. Those 3 things. That's the simplest way I can summarize it for you. Don't worry, I'm going to go through human evolution. I do see everyone asking about it. It's going to be what's after genetics, because I know people wanted me to do genetics. Tegan says, ma'am, you said different protein, different amino acid. Oh, I said different amino acid, different tRNA, different protein. Those were the 3 things that I had mentioned that you would get marks for. What's the difference between transcription and translation? Guys, if you're asking me at this point, I'm a little bit nervous, so we don't know the difference. Translation is when you are copying the message. No, what am I saying? I beg your pardon. Transcription, to transcribe, means to write. So transcription is when you are copying the code, and translation is when you are reading it and making a protein. Someone wants me to repeat the history of DNA. Sure thing. So here we go. DNA was discovered to be a double helix shape by Watson and Crick, and they used X-rays. A lot of people think that there's more to it, and there isn't. That's it. I know that we talk about Rosalind Franklin a lot too, maybe in your classroom, as the original or the real discoverer of DNA. But unfortunately, they haven't changed the guideline to reflect her in the discovery of it. And to answer the last group, yes, I am going to be posting this when we're done. Okay, so I think what I want to do is move on. Otherwise, we're going to run out of time, and I haven't covered everything I want to cover. Okay, so let's move on to genetics. Okay. Genetics is a passion of mine. It's something that I mark every year at the end of the year. Just by chance, I always get the genetics question. So I'm telling you the things you keep doing wrong that you end up losing marks. And this first one is going to sound very obvious, but a lot of people don't get 6 out of 6 for their genetic cross layout. So what must the layout always look like? So to get full marks, we always have to say P1 in the column or in the side column of your page. You are always going to have to put the parent phenotypes first times the other, and I'm just going to put here PP for parent phenotype. Below that, you're then going to put the parent genotype times, and I'm just going to put here PG as in the other parent genotype. You are always going to write the word meiosis below that. In the column over here, gametes. And now what I like to do is I like to put circles around my gametes, and to show you this more clearly, I'm actually going to do an example of parent geno and phenotypes. So let me do a classic one over here. Let's say it is a black cow times a white cow. I'm going to make them unusual, and then I'm going to make this a big letter and a small letter and 2 small letters. Now, without me doing any more work, I'm hoping that everyone can see what the letters mean and who is the dominant color and who is not. Remember that if this is complete dominance, the moment there is a capital letter like that, it tells you what the dominant color is. So clearly black is the dominant color, because in order to be white, I've got 2 small b's. That means white must be recessive. So I didn't even have to do a key or an explanation. You should be able at this point to pick it up. The moment you see a capital letter in a complete dominance, you should know that that is the dominant color. So when I draw my gametes, your teachers probably don't teach you to do this, and that's okay. I just do it because it prevents my students from losing marks. I make them put their gametes in circles, sort of like to represent cells. And why do I make them do that? Because if they don't, often they lose marks for putting the gametes too close together, which is problematic. So we put our gametes into separate little circles. I then write the word fertilization, and I use a Punnett square. I don't know what your teachers have taught you, maybe a line diagram. I don't particularly like line diagrams. They are problematic for many of us. We make too many mistakes. I love a Punnett square, so I prefer Punnett squares. And I'm going to transfer my gametes in. So remember, you've got to keep the maternal with the separate from the paternal. In other words, what came from one parent must stay in one column or row. So I'm going to put the big B, little b. And for the mom cow over here, I'm assuming it's the mom. It doesn't be the mom. It can be the dad too. The 2 small b's. Right, now we multiply in. So we've got big B times small b. This b times that gives me 2 small. Big B times small b, 2 little b's. Right, now we need to write out, and I do this as an extra step. Why, ma'am, do you do it as an extra step? Because it guarantees that nobody gets any marks lost, and that's what I'm all about in my classroom, making sure that nobody makes a silly mistake that would end up making them lose marks. I foolproof it, is what I'm trying to say. Is it a little extra writing? Yes, but if you do it my way, you won't ever get it wrong. So let's write out the percentages of the genotypes. So all the matching genotypes, if we put them all together, this one and this one are the same genotype. So I've got 50% big B, small b, and these 2 joined together, they're the same. So I've got 50% 2 little b's. And if they ask you to write a ratio, that would be a 1-to-1 ratio because it's 50-50, right? The phenotype, right, in other words, the physical characteristics of these cows, well, I've got 2 that have 2 capital B's, which means that I've got 50% black cows, and I've got 2 with 2 small b's, so 50% white, and again the ratio is a 1-to-1. The ratios are important in genetics because they tell us, or a ratio can tell us, what the parents were. It's a little bit more complicated than I'm making it sound, or maybe it sounds complicated, should I say, and I'm not elaborating more on it. I have made a video on this and ratios. If you want to go and watch my monohybrid and my types of dominance videos, there are 2 in the grade 12 playlist. Those will really help you out a lot when it comes to doing these crosses. So let me show you, if this was your page, how this would be marked, right? So first things first, 1 over here for the 2 parents, 1 over here for the parents' genome, genotype, 1 over here for the gametes. Meiosis and fertilization go together, so that's another 1. Writing your genotype out in your little cross here, that's another 1. So 1, 2, 3, 4, 5. And now these answers here, generally it's based off the question. I give both because it's foolproof, then you'll always get the marks. But sometimes they'll be like, what is the genotype percentage? Well, then the mark will go here, right? Or they'll say, what's the phenotype? Then the mark will go here. Another optional mark, by the way, is this one over here. So actually you have 1, 2, 3, 4, 5, 6, 7, 8 possible marks. And generally what I like to say to my grade 12s is, if you write this out and it's out of 6, let the marker select the 6 of the 8, right? Don't think for the marker. Don't say, well, I'm just going to make them focus on these ones. Rather, do this outline, and then you will always get 8 out of 8. So that is the basic genetic outline. I have seen there's a whole bunch of questions, so I'm going to have a look at the comment section now quickly. I have a question. When do we answer in the positive and when do we answer in the negative? It's a great question for the exam for tomorrow. You always answer in the positive, unless the question is saying anything like when it doesn't happen, when it's not working, when there isn't X, if it's damaged, if it's broken, if there's a mutation. Those are all negative answers. Negative answers mean take your original positive answer and explain it if it was broken, missing, changed, deleted, removed, something like that. That is the negative answer. All right. Let's see. Sorry, there's so many comments. It's taking me a hot second to read through everyone. Everyone wants maybe to explain the Punnett square again. I can, because I'm going to do another cross. Dihybrids. Somebody asked me, is this a dihybrid cross? No, friends. This is not a dihybrid. This is a monohybrid. Let's see. Gametes have to always be apart. Yes, so like this. That's why I put them in circles. We don't want them close. Types of dominance, types of dominance, types of dominance. I see all of that crossing examples. Okay, so let's just touch on that before we go on to anything else. Okay, so you get 3 types of dominance, right? You get complete dominance. You get co-dominance. And you get incomplete dominance. And I'm going to do some examples of these crosses using these 3. So complete dominance is when you have a capital letter and a lowercase letter, and they have contrasting colors or characteristics. Like this would be black would be the dominant color and white would be the recessive color. And we use big letters or small letters to tell the difference between the one that is dominant and the other one that is recessive. And so there's a law that governs this, and please know the laws off by heart from your guideline yet again. Co-dominance is when you have equal, I'm just going to put here as a little reminder, this is when characteristics are equally dominant. And so an example of this and how we annotate co-dominance is we would have like, let's do, I want to think of a good one now. Let's do a classic one like red and white. Now you'll notice that there are 2 capital letters this time, different from here. You'll notice that the letter matches both of these characteristics because again, they are equally dominant. So they each get their own lettering separate from one another. And co-dominance is if you get RR, this flower will be red. If you get WW, it'll be white. But if you get RW, it will make red with white spots, as an example. Co-dominance is also seen in blood groups. I'll touch on blood groups very quickly. So don't worry. Sorry, I'm just pausing because there is a comment block. Just remember, everybody, my comment section is not a place to argue. So YouTube picks that up and blocks you. So just remember that, okay? So this is codominance where they're equally dominant. So it literally means we could have like a red flower, and on that red flower, yes, there are red spots, but there also could be white spots. I know you can't see the white so clearly on here now. You know what I mean? So if I zoom up on there, you can see my white spots on my red flower. It's a little bit hard to see. But that is when you have codominance, where they're equally dominant, right? But what about incomplete dominance? Incomplete dominance is often confused with codominance. And I understand why. Let me show you why. Incomplete dominance is when there is... Oh, you can't see me right with the white pen. Incomplete dominance is when there is a mix. Let me use red and white again, and let me show you why it becomes confusing. So again, you still use capital letters, red for red, white for white, so W for white, except this time, you still get RR makes red, white, white makes white, but an incomplete dominance, so this still makes red, this still makes white, RW will make pink. And so incomplete dominance is when there is a mix of the two. Again, the lettering that you use is often the reason why we get marks taken away and we get lost. Let me show you what I show my pupils. I'm going to get rid of this quickly. And I'm going to show you what I teach them. It helps a lot. Because codominance is when they're equally dominant, what you are allowed to do is you are allowed to use the letter C for co, and you superscript the R and the W on the top. So now this still means red, and this still means white. But when you write them out, you now write them out like this. So this is still red, this is still white. And if you go CW, that is red with white. So red with white spots, for example. This lettering over here is acceptable in an exam as long as they haven't given you the lettering. In other words, if they give you the lettering, use what they give you. If they are allowing you to create your own lettering, you're more than welcome to use this kind of annotation for codominance in particular, so that you don't confuse codominance with incomplete dominance, because their letterings can be very similar and we get caught up in the moment of how to calculate their crosses, right? So I would do that. These are the three options you can use depending on the type of dominance. The only way to get good at these kinds of crosses is to do many of them. And I do have quite a number of genetic videos in my grade 12 exam question playlist that you can go and watch. And I walk you through how to do these. So I suggest you go and do that, particularly everyone. And I'm all directing you to this one particular video. If you go into the playlist and you scroll through the thumbnails, look for the genetic one with the snails on it. That one is a really good question from a past paper. It is a hard question, a very advanced question. And the reason why it's advanced is because it combines both complete and codominance at the same time. So it's a lovely advanced question, like worst case scenario that you might get. Okay. Now I'm going to pause there for a little bit. Some people are saying they are lost. You need to be a little bit more specific when you say you're lost, so I can't help if you're not specific. Lillian is asking, ma'am, will WR then make white flowers with red spots? Yes. Yes. So it doesn't actually matter the order, if that's what you're asking me, Lillian. I just put red with white, but it could also be, yes, white with red. In other words, the order in which they go together is up to you or up to the way they've phrased it in the question. Okay. I think what I want to do to make sure that we stay on track is just go over blood groups quickly, because I know we're not so great at that. And then we will do evolution. I promise. Promise, I promise, I promise. That'll be my last section to do anyway, would be evolution. So just a quick recap. When we talk about blood groups, we use the letter I, right? And when we use the letter I, we superscript it with either an A, a B, or a lowercase I. Those are the three options. So if I have blood group A, I'm either this or this. Same for B or this. You may only use this annotation in exams, everyone, nothing else. And if you're blood group O, then you're two little i's. So this is blood group A, this is blood group B, this is blood group O. And I can't forget, of course, blood group AB, which is this. So this is AB blood group, right? So blood groups show us an example of co- and incomplete dominance happening at the same time. Let me show you what I mean. This, oops, let me just close that. This is an example of co-dominance because A and B are equally dominant to one another. Remember, if I go back here, see? Equally dominant, co-dominant. So AB blood groups are co-dominant. But if we go to the others, you will notice up here that when we put an A blood group with an O or a B blood group with an O, they end up just being AB. And that is because this grouping together creates complete dominance, which means one is always dominant over the other. In other words, A is dominant to O and B is dominant to O. Now, the most common way they ask this is something along the lines of, the parents are this, the child is that, is this their child? Or is this child adopted, etc. So let's say they give you a question where mom is this and dad is this. Let's say mom is blood group O and dad is blood group A. The child is O. Is it possible that this is their child? Now, before you all say, yes, 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 because mom is O, I need you to pause for a second there and not just make that assumption, because I can do a different one where the parent is something, but it's not necessarily their child. And I'll show you an example of that. Because mom is blood group O, she can only be two little i's. Because the child is also O, they must also be two little i's. So now, what is the dad? Now remember, the dad can have two options. He can be two capital letters for blood group A, or he can be A with a little I. Now, according to the law of segregation, which is why we must know our Mendelian laws from Mendel, to make this child, in order to make this child, mom can only give one I, which is this one. And now, which pair is dad? Is he AA or is he A little I? Well, to make this child, if mom gave this allele here, then that means that dad must be this pair to give this little I. So yes, this is their child because this is mom, this is dad. I've highlighted what they gave their child. But let me show you the example I wanted to say now where you look at mom and dad and you think, oh, automatically it must be their child, but that's not for certain. Let's say we have a child. Let's say we have a parent that is A. So mom is A. And dad is AB. And the child is O. Again, let's not look at everything at face value and go, oh, automatically, because this doesn't match up, this can't be their child. Let me do a different one, actually, because I want to prove my point a bit further. Let's make this child a AB. No, do I want to do AB? No, because we can still make that possible combination. Let me think of one. Actually, let me change both the parents so I can show you when it's not the same. Let's do a different one. Let's go with mom is AB, dad is O, child is O. Okay, so you look at that and you go, sometimes your brain goes automatically, oh, well, dad is O, child is O, it must be his child. But hold on, let's actually do the calculations here. Mom can only be big A, big B. That's all she can be for AB blood group. Dad is O, which, as we heard, is recessive earlier. So he can only be 2 little i's. Now, this child is also O for 2 little i's. But here's the problem. If we follow the law of segregation, one of these alleles came from dad, potentially. But this other allele, it can't come from this mother. This mother only has an A or a B, which means that if this was her child, this should be a child that's either A or B, but it's not. So now what's interesting here is this child cannot be from this mom, because this is what she has to offer, and this child is carrying neither of those alleles. And this is potentially the dad, but not necessarily the dad, even though they have the same alleles here. That's not a confirmation of paternity. So what we have here is sometimes this would show that the child is adopted, or this child is this dad's child, but this mom is not their mom. It's their stepmom or their adoptive mom. So you've got to make sure that their alleles match one another. Okay, so I am going to just look at the comments, and then I'm going to move on to evolution, because I know that's probably your biggest concern for tomorrow. Somebody said here, blood groups can't determine paternity because the suspect's parents can have the same. Yes, so by the way, blood groups can never determine paternity, which means if we look at this over here, you can't confirm a father. You can only exclude fathers, because there are too many men with the same blood group. Just because you have the same blood group as a child doesn't mean they are your child, basically. All right, everybody. Cloning, evolution. Somebody says, will we see Australopithecus and Ardipithecus tomorrow? Potentially, yes, you will. Everyone says human evolution, pedigree diagrams. Ooh, I don't know if I can fit it all in one live lesson, guys. Okay, I'm going to be like super fast, quick, fast. Here we go. Cloning. Okay, I did this in the video the other day, but I think that was our members only live. Okay, quick, fast, cloning. Last thing I'm going to do on genetics, and then I must move on, because I know a lot of you overlook cloning and you're all requesting it. So, okay, we've got sheep A. We've got sheep B. Out of sheep A, we take an ovum. Out of sheep B, we take a somatic cell. If you don't know what a somatic cell is, it is a body cell. It is a cell that is diploid. So this is diploid, this is haploid. For the ovum, we take the nucleus out and we end up with an empty ovum like this. It's called a gutted cell. In the somatic cell, we want the nucleus because that's where all the genetics is for the animal we want to clone. So we get rid of the cell and we're left off with the nucleus. We put the two together. So we put the nucleus inside the gutted cell. We give it a little electric shock, which stimulates mitosis. Oh, I'll leave it there for mitosis. So it stimulates mitosis. And we now end up with a zygote. And my final question to you is, if I'm cloning, who am I cloning? Am I cloning sheep A or sheep B? The answer is whoever the nucleus comes from. So in this instance, the nucleus came from sheep B. So this will be a zygote of sheep B, aka a clone of sheep B. So that is reproductive cloning. They can then say, well, then who is sheep C? Sheep C can be a surrogate. In other words, you put this zygote into a third sheep, like a surrogate mother will carry the baby at the end. Okie dokes. Right, I must move on, otherwise we'll never get to evolution, and I know a lot of you are here for that. I know some of you have also asked about pedigree diagrams, but I'm not going to have enough time to get through pedigrees as well. I have so many lovely videos on pedigree diagrams, everyone, that I suggest you go and watch. There are also other lives from back, like if you watch backwards, other lives I've done pedigrees. Always look at the thumbnails because I include the topics in the thumbnail so you can see what we did in that live based off the pictures. Okay, evolution. Here we go. So we have something called natural selection, which is also known as Darwinism. And natural selection is that whole explanation that you should be taking out of your exam guideline, everyone. There's like literally a bullet point, step by step, of how you explain natural selection. But the key is that when you talk about natural selection, you are talking about favorable versus unfavorable. So a classic example I did with my members the other day in a video was, you have a pond and it's filled with green frogs and yellow frogs. They're all the same species, by the way. They're just different colors. And let's say that the pond is also filled with green algae, which means it's favorable to be green. It is unfavorable to be yellow because you're more easily seen by predators. And so going with this explanation from the guideline, from the guideline, what you need to do is substitute these facts in. So the green skin, so first of all, you'll say there is variation in color, which there is. And you're going to say some are favorable, the green are favorable, the yellow are unfavorable. If you are green, you're going to survive, you're going to reproduce. Oh, I'm not spelling. Produce. Whereas if you are yellow, you will die, you will not reproduce. And if you are green and survive and reproduce, then there will be more green frogs, or there will be more frequency of the gene. So that is natural selection, right? Natural selection, I also like to think of it as a tool of evolution. It selects what is and is not favorable. Now remember, this is in a species, everyone. This is not one species of frog and another species. It's just frogs of two different colors. Same species, just like humans. Same species, just different colors. How does this become another species? Okay, so let me give you the explanation for speciation. Again, we must take speciation from the guideline. Please, everybody, please, 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 please. I don't want you using any textbook, anything else but what's in the guideline. If you have my study guide, I use the guideline explanation as my explanation. Okay, so you can trust what I have written there, but you cannot always trust what is written in textbooks. Please use the guideline as your answer. So speciation. We talk about geographic speciation, which is when you separate organisms from one another through a barrier. The most classic example is we have a piece of land and it gets divided. Let's say, for example, I had lizards. On this side, again, I'm going to use the word color green. So I'm just going to do this. Here's my green lizards all over the place. And then I'm going to do, let's do brown. Okay, we've got brown lizards now. So there's some brown lizards. There they are. Okay. So what happens is, here is my little area of land, and let's say a mountain range forms here, right? So this is my mountain range. There it comes, dividing the population into two. Okay. So now what we're going to do is we are going to explain speciation, and we're going to mention that there is, again, variation in a population. In this case, we've got brown lizards and we've got green lizards. We are going to mention that there is a geographical barrier. If they tell you the barrier, please use it in your example. Here it's a mountain, a mountain range. Okay. From there, so geographical mountain range, from there, what we are going to need to include is the fact that the two populations experience natural selection independently. And please, everybody, this is the only time you mention natural selection here. Sorry, everybody, we just got disconnected there for a hot second. No, I'm back. I'm back. Sorry, everybody. We just got disconnected there for a hot second. No, I'm back. I'm back. Sorry. I don't know why I just disconnected there. I beg your pardon. I don't know what it was, but we're back. Don't worry. Don't worry. Just a little flicker. Okay, so just a little flicker. Okay, so two populations experience natural selection independently. I have not spelt independently correctly, but we move, okay? So they experience natural selection independently. What does that mean? Well, let's look at our drawing over here. Let's say this side of the mountain range is greener, which means it's an advantage to be a green lizard. It's greener, it's lusher, there's more vegetation, there's more food, you can camouflage nicely. On this side, it's more drier and brown. So this side, there is more vegetation. Here, it's more sandy on this side of the mountain range, okay? And so what happens is if you suit this side of the environment through natural selection, you'll be green, you'll survive. If you suit this environment, brown and sandy, you will survive. And so you experience natural selection independently. Now, here's the key bit. They become so different that if they were to ever mix again, right? So if we ever took a brown lizard and a green lizard and we tried to mix them again and we tried to make them interbreed, they would be unable to interbreed and produce fertile offspring. Now, I know you're probably thinking, but ma'am, aren't we just changing their colors here? No, we're not just changing colors. I do know something is wrong with the connection. I don't know why there is no sound. I wonder if there's no sound still. Just let me know, everybody. Can you hear me? Give a thumbs up. Or a yes, we can hear you. Somebody says they have sound. Don't worry, the video is lagging because for some reason, I don't know why actually it's lagging. There is sound. Okay, good. Okay, great. So I know you're probably thinking, but ma'am, they're just like green and brown, okay? It's not about just being green and brown. These lizards on this side are not just green. They are green, they can camouflage, maybe they've gotten bigger, maybe they eat different food, because remember this mountain range is blocking off their reproduction group. They can't reproduce with each other. So if you're on this side, you can reproduce with all the green individuals, and maybe you eat specialized kind of food on this side. Maybe if you're a lizard who got stuck on this side, a brown lizard, you are more suited to this side because you can camouflage better in the sand. There's less vegetation, which means there's less food. Maybe you become an insectivore on this side. So maybe our green lizards are vegetarians now. Our brown lizards are insectivores. And when you accumulate so many of these differences, when they become so different, they're unable to reproduce with one another because they live in different places. They eat different foods. They might be different sizes. Maybe the green lizards only come out at nighttime and the brown ones come out during the daytime now. Because of these differences, if they were to ever mix again, they'd be unable to interbreed and produce fertile offspring. So that's what makes species different. And we're always talking about the phenotypic differences in these instances. They very rarely ask for genotypic differences. There was a question about artificial selection earlier. So you know the explanation for natural selection, and I talked about favorable and unfavorable. If you have to do an explanation for artificial selection, I would substitute the word favorable for desirable. So if we were talking about like trying to artificially select cows that make a lot of milk, I would say I would select the desirable cows that make a lot of milk versus the undesirable cows who don't make a lot of milk. I would not let them reproduce. So artificial selection is exactly the same explanation, except you use the word desirable instead of favorable. And instead of saying letting them die, you just wouldn't let the desirable ones reproduce. So there's nothing wrong with the undesirable ones in terms of their health. They just don't make enough milk. So you don't want them to continue reproducing. Okay. So we've got evolution, speciation. The last thing I'm going to wrap up this whole thing is off with it. Sorry. The last thing I'm going to wrap this up is with human evolution. Okay. Again, I've got a lovely section on this in my guide, in my study guide. I simplify it so easily in the study guide, but also in the videos I've made for you guys too. So please also use those as a resource. But when it comes to human evolution, the main focus you should be looking at is the fossils, right? Now, before you say, are you talking about Australopithecus and all that, ma'am? No, I don't mean that. What I mean is, if you're given a skull from the bottom, and they're saying, here's the foramen magnum here, and here's the foramen magnum here. Just write it on the side here so you know the word. Which one of these two is bipedal? Is it A or is it B? When we are talking about the foramen magnum and looking at fossils. So if we're looking at these two fossils, hopefully we know that the foramen magnum tells us it's bipedal and that B would be our bipedal organism. Oh, oops. Why is it bipedal? It's bipedal because the foramen magnum is more forward. Please, everyone, do not use the word central. No, it's more forward only. Do not use this word. If you want to talk about this one is more back. But please, no central, nothing, okay? Another thing, if they're asking about bipedalism, is the shape of the pelvis. So again, is the pelvis, and I'm just doing a very generalized shape of a pelvis. So this is the pelvic bone versus this pelvic bone. Obviously, this is not what a pelvis looks like, but I'm trying to show you the difference. This one is short in terms of it's short this way and broad, which means this way. This one is long this way and narrow, which means this way. So between A and B, out of these two fossils or these two pelvises, which one is the bipedal organism? Hopefully, everybody is going to say A. Why? Because a short, broad pelvis holds up the entire body weight in an upright structure. That's what is linked to bipedalism. So these are the two main things that are linked to bipedalism when you're explaining them. The foramen magnum, the pelvis. Another thing that is directly related to the bipedalism is the shape of the spine. Is it an S shape or is it a C shape? If it's an S shape, then that organism is bipedal. If it's a C shape, the organism is a quadruped. It means it leans forward and walks on in its hands and feet as well. Okay. Let me think of something else that they will ask in terms of this. They sometimes ask about teeth. So big canines versus small canines. Remember, humans have small canines because we chew less, we tear less, we cook our food. I think the big thing that a lot of us need to know is how the changes in the skeleton are linked to the changes in our intellect, into our intelligence. In other words, when we look at a skull and we look at I'm just going to quickly emphasize what I'm doing here. And we look at a skull of an Australopithecine. So like this is the Australopithecine skull, right? When you are looking at their skull, we've got to look for a couple of things that are linked to their intelligence. And is this us? So what are the things we look for? Number one, actually, I've drawn this a little bit too big. Their skull is not this big. Their cranium is not that big. They've got a very big jaw. So let's add in the lower jawbone properly. And that's their skull in the background. Okay. So if we're looking at their skull, number one, they have a small cranium. That's because they have small intelligence, small brain. They have got a brow bone or a brow ridge. They have a sloping face. They have large canines. And they have this thing over here, which is called a prognathous jaw. Now, what's interesting about the prognathous jaw is it reveals many things. If you have a prognathous jaw, you generally have a sloping face. You generally have no chin. I know you're looking over here and you're like, man, that's a chin. No, it's not a chin. It's not a chin. It's not a chin. No chin, sloping face, large canines. These things that I'm mentioning to you here, these things, let me highlight them. Brow ridge, sloping face, large canine, no chin. They are all associated with the prognathous jaw. The prognathous jaw creates these things. Okay, these are the things you are looking for. And you use these things to identify where the skull is in relation to the others. Generally, you will get 2 or 3 skulls, and they'll be like A, B, and C. They'll be out of order. okay? And they'll say, place these in order from oldest to newest, or which one is the chimp, or which one is the Australopithecine, which one is the human. You are going to look for these characteristics, these things. And if it's a small cranium or the smallest cranium, it must be the oldest organism. If it's got a big cranium, it's the newest one. If it's got a small jaw, it's the newest one. If it's got a massive jaw, it's probably an ape or a gorilla. Does it have a sloping face or a flat face? Well, everybody, if you're not sure what a flat face would look like, literally it goes down like that, and that's where our nose would be over there, and this is our mouth. So this is a flat face versus a sloping face. Okay. Again, I have a great video on this that I think everyone should go and give it a try. The last thing I'm going to say about human evolution before I sign off on this video is you must know your fossil names, please. So Mrs. Ples, Karabo, Taong Child, you need to know their names, as I just said now. You need to know who discovered them. You need to know where they were discovered. So Tanzania, Ethiopia, South Africa. And lastly, you need to know what example of what species are they. So what was Mrs. Ples? What was the Taong child? What were they? Were they Australopithecus africanus or Australopithecus afarensis? Okay. And I think that's where I want to leave it. We've had a long lesson. This is a really long live lesson. It was an hour and a half. If you've missed anything because you've just joined, the video will go live shortly after I end this live. It'll upload immediately. We've covered everything from meiosis to DNA to genetics to evolution and cloning. So we've covered a lot in this hour and a half, but I encourage you, if there's anything I was unclear about, to go and look at my grade 12 playlist, go and watch the videos on the topics tomorrow, and also please go and watch the videos where I go through past paper questions. Okay, thank you everybody. I hope that this has been useful to you. If you are a member of my channel, specifically the Rescue Me members, it's different to subscribers, everyone. Then you can get access to my study guide for free. You just need to find it under your perks info in your community page. Otherwise, if anyone is interested in buying the study guide, it is on my website, missangler.co.za. Okay, thank you everybody. Hope that's been helpful. Good luck for tomorrow, and I will see you all soon. Bye.